Hi, and welcome to Chapter 9, where we are going to learn all about the cell cycle. And most of you, when you think about the cell cycle, you tend to think about this term mitosis. And it does turn out that mitosis is part of the cell cycle. So let's go ahead and dive into the key things that are happening in cell division and really get an overview of mitosis. So to start out with, it's very important to realize that organisms have to be able to produce more of their own kind, and that is what one of the key factors to distinguish living things from non-living things. So ability to reproduce. And the way that life reproduces is by reproducing at the level of the cells. That's actually a really important concept because a lot of us, when we think about, having another generation, having babies or whatever, we think about just the act of sex or making a new living thing that's separate from ourselves. But that is not what happens with any kind of reproduction. All reproduction happens at the cellular level. So we'll talk about cell division. Now, I know I was just talking about sexual reproduction, and that turns out that that is not one of the things that we're going to be covering in this chapter. That's the next chapter. So what is it that we're going to be talking about this chapter? Well, one of the first things that we're going to be talking about is this concept of asexual reproduction. That means where you reproduce, but it does not involve the act of sex, or we're not even talking about like how we think about humans. We're talking about like not exchanging information, genetic information amongst organisms. You're just dividing. And this is how many organisms like amoeba reproduce. They do it basically just by splitting themselves in two. And since they're single-celled organisms anyway, this makes sense that that would be the way that they would reproduce. For multicellular organisms, one of the main key roles or functions of asexual cell division, like that happens inside our bodies all the time, is growth and development. You can't get bigger unless you have more cells. Your body can't change as you develop into a, grown up, basically, or as you go through major changes in your life. That's called development. And development cannot happen unless there's cell division. So cells become more and more specialized, those kinds of things. The third thing that asexual or basic cell division that we're going to talk about today is tissue renewal. And this happens when, let's say that you need more blood cells. Well, your blood cells are made in your bone marrow, and those new red blood cells are being replenished and replenishing old used up ones. So tissue renewal is a big deal here. We like to say that single cells can reproduce asexually, so can multicellular organisms at the level of the cell. Once an organism is fully grown, the same cell division processes that helped you grow also renew, repair, and replace. Renew, repair, and replace. Those are the 3 R's of cell division. Especially the specific type that we're talking about today, mitosis. So cell division is integral part of this thing called the cell cycle, and we're going to hear all about the cell cycle very shortly. But you should know that cell division is only a small part of the cell cycle, and it's essential, again, but it's only a part of the cell cycle. Our first big concept is that most cell division is going to result in genetically identical cells. So important. We want to have DNA that's exactly the same in both cells, and we call those daughter cells. That two end cells are called daughter cells, as you see right here. DNA is going to be passed from one generation of cells to the next generation of cells with something we say is with fidelity. And that just means that they do a pretty darn good job of copying them. So they copy with fidelity, or a good job, an accurate job. If we start thinking about the DNA inside of a single cell, all of that DNA inside of a single cell is known as the genome. You might have heard of the Human Genome Project. And with that project, they literally took all the DNA from a single cell and they figured out the sequence of A's, T's, C's, and G's. But genome is an important word when we're thinking about things like cell division. A genome varies depending on what type of organism you're talking about. So for example, a prokaryotic cell like a bacteria or an archaeal cell, those only have one single molecule of DNA. But you and I, we have 46 separate DNA molecules all located inside of our nucleus. And so things get a little tricky when we start to recognize that there are 46 chromosomes. Now, there are different numbers of chromosomes in different organisms. 46 is not a magical number. It just happens to be the number that's found in humans. When we think about the DNA molecules themselves, we say that they're packaged into these structures called chromosomes. And you can see some of those chromosomes, like these unique chromosomes right here. Here's one that seems to be kind of U-shaped in the cell. Here's one that looks a little squiggly here, right? Those are chromosomes, and those chromosomes are made up of DNA and some other things too. Looking a little bit more closely and thinking about DNA and chromosomes, when we're talking about eukaryotic chromosomes, remember we have 46 of those, right? And we are eukaryotic cells because we got the nucleus. So the chromosomes are consisting of two basic things. Sorry, let me just back up a second and say that chromosomes consist of something called chromatin, and together with DNA and some proteins, that's what makes up the chromosome. So chromatin is a complex of DNA and proteins together. And when you got that, you can say it's chromatin. Now, we use the word chromatin very rarely when we're talking about DNA. But when we're talking about cell division, chromatin is a key phrase. And so this idea that it's chromosomes or chromatin, proteins all together, that's really key. I already talked to you all about how every eukaryotic species has a very specific and characteristic number of chromosomes in their nucleus, and we have that 46. But what I didn't say is that the 46 is actually two sets of 23 chromosomes. So, and we'll get a lot more information about what I mean by sets of chromosomes when we get into chapter 10. But for now, it is an important concept that you keep in mind that you get half of your DNA from your mom and half your DNA from your dad, and that DNA is very, very similar to each other. I mean, mom and dad, they're both humans, they're both very, very similar to each other. So they each give you 23 chromosomes, and you have a total of 46. Now, if we're looking at your body's cells, we're going to call your body's cells by a very specific name called somatic cells. That's the word right here, somatic cells. And soma actually means body. So somatic cells, those are any cells in your body that are not reproductive cells. So we cannot consider things like gametes. Those are not somatic cells. Even though they're in your body, we don't call them body cells because they are different. Those are reproductive cells, your sperm and your eggs, and those have got the number of chromosomes cut in half. So the gametes, the sperm or eggs, the reproductive cells, they only have 23 chromosomes for humans. Again, whenever any species is going through reproduction, they are going to cut the number of chromosomes in half when they make gametes for sexual reproduction. Not everything reproduces sexually, but that's what we're focusing on. So let's take a closer look at what happens to chromosomes during eukaryotic cell division. And the first thing is that when a cell is not dividing, the chromosomes are long and thin, and basically they're in this phase or stage or kind of view that we use this term chromatin. So chromatin is going to be long and thin, and we can say not condensed. The picture at the bottom of the screen, that's not the chromatin form. Chromatin is, you can think about it as like spaghetti. It's all the spaghetti, in through your nucleus. But when you're getting ready for cell division, the DNA does a couple things. First of all, it replicates itself. And also, the DNA condenses or thickens up, it shortens up, it curls all up, and it becomes something that we consider to be a chromosome shape. So what you see right here is a very condensed chromosome, not the spaghetti like chromatin, not chromatin, but a condensed chromosome, and it actually has two parts to it. Each part of this is called a sister chromatid. Now, look, there's nothing I can do about the language here. The names are the names. So we've talked about chromatin with an N at the end, and now we're talking about chromatids, and that's a different thing. And we'll see what this happens, as we're going along. But this part here is an identical copy to this part here. We say they're duplicated. They're attached at this region to each other. They're literally attached to each other at this region called the centromere. And so these two identical chromatids, sister chromatids, are attached at the centromere. So that's your basic structure of when we look at chromosomes during cell division. That's what we're going to be thinking about. But again, remember, it starts out as as chromatin, these long spaghetti fibers, and they shorten, they coil, and they condense up into this chromosome shape where you can really see. Kind of looks a bit like an X shape if you think about it. Use your imagination a little bit. Turn your head on the side, and you can see a bit of that X shape where here's one half of the X and here's the other half of the X. So let's get some understanding, and this is again a very basic understanding of what happens during cell division and how chromosomes make copies of themselves. Now, we are not getting into any details at all right now about how the DNA makes copies of itself. We'll say that for another chapter later. For now, we are going to just kind of say, snap your fingers, and you go from having a single unreplicated chromosome to a single replicated chromosome with two sister chromatids. We still count this as one chromosome because it's connected at the centromere, and so it's literally one molecule. So this one's unreplicated, and here it is replicated, or we might say duplicated, consisting of two sister chromatids. When we go through cell division, these sister chromatids are going to disperse or separate into separate cells. So this one went into this cell, and the one over here went into this cell, and voila, you have two exact copies of what the original cell looked like with regards to its genetics. All right, so again, just focusing here on eukaryotic cell division, there are, and we're going to talk about the cell cycle too, but within this concept of the cell cycle, we have the division process, which is called mitosis. And this mitosis process, it really just focuses on what happens to the genetic material. But of course, the cell itself has to separate as well. And the word that we use to describe how the cytoplasm separates. That's called cytokinesis. So mitosis and cytokinesis are kind of, they go together hand in hand, but they're different. Mitosis focuses on the genetic material, the stuff in the nucleus, and cytokinesis focuses on what happens to everything else in the cell, the cytoplasm. Just a reminder, the gametes are produced by a different kind of cell division that's called meiosis. We'll cover that in the next chapter. And the main thing that I want you to keep in mind as we're going through talking about mitosis is that it is different from meiosis in many ways. But the big one is that meiosis is going to end up giving you non-identical daughter cells that have only half of the amount of chromosomes. We say one set of chromosomes, half as many as the parental cell. Whereas mitosis, you're going to get the exact copies. So let's start to take a look at what happens in the cell cycle. I've been alluding to the cell cycle for a few minutes now. And this is a good picture of the cell cycle. I like this. Looks like a pie chart, right? The cell cycle is divided into two basic phases. The teal phase right here, which you can see lasts a very long time. If this is a pie chart, lasts a huge long time, compared to the second phase in the cell cycle, which is called the mitotic phase. And we give the mitotic phase the name the M phase, just for short. The M phase or mitotic phase, you can see, is divided into two subsets, mitosis and cytokinesis. Remember, mitosis focuses on the nucleus and the genetic material, and cytokinesis focuses on the cytoplasm. But what about that interphase thing? That takes up the vast majority of the cell cycle. And it is actually subdivided into some parts as well. So there are three parts to interphase. G1, S, which is a shorthand for DNA synthesis, and G2. Actually, G stands for gap. So literally this is the gap 1 phase and the gap 2 phase here. And the reason it's called gap is because when the scientists first started looking at cell division and they saw this, under their microscopes, they saw it like nothing seemed to be happening. So they just called this the gap phase. There's a lot of stuff happening during the gap phases. A huge amount of stuff is happening during the gap phases, but not that much of it is important to cell division. So while tons and tons of really important things are going on, like all the metabolism that you're ever doing is mostly happening during the gap phases, not so important for focusing on cell division. So the thing that we focus on when we think about cell division is the S phase, or the DNA synthesis phase. And so that's super important because if DNA doesn't make copies of itself, then you're not going to be able to divide that DNA properly and get identical daughter cells. So again, we're going to focus on the S phase when it comes to interphase. When it comes to mitosis, there are, we in this textbook, we're dividing mitosis into 5 phases, 5 subphases. Just so you know, not all books do this. Usually it's prophase, metaphase, anaphase, and telophase. But your book very properly talks about prophase and then something called prometaphase, then metaphase, anaphase, and telophase. So it's PPMAT. If you're following along and trying to come up with a good mnemonic, that's always a good idea. So, and remember we just talked about cytokinesis, and with cytokinesis, that's the cytoplasm happening, dividing, and that is going to be overlapping in time with telophase. Let's take a look at some of the things that are happening in interphase a little bit more carefully, and then we're gonna kind of scooch into the concepts of mitosis and those subphases that we just talked about. So Remember that DNA makes a copy of itself during interphase. It's so important that you remember that it's happening during interphase. So remember, we go from having one chromosome to having, that's supposed to be a little arrow right there. We'll make that a little bit of a better arrow for you, to making a replicated chromosome. And that happens during the S phase, right? In G2 of interphase, we're going to have some important stuff happening, like growth and all these important metabolic things. But this is just a setup for you so that you can kind of see what's going to be happening to us as we go into mitosis. All right, so diving into mitosis. The first thing that I'm going to say to you is that in prophase, you got a whole bunch of stuff going on. In prophase, look at what's happening. There's this structure right here, and we'll get some more details on this structure in a second. This is called the centrosome. And you don't see that word on here right now, but you do see the word aster. And so what we have here are two centrosomes with their aster structures. We have these things called mitotic spindles. And so what's happening is these centrosomes are starting to split apart or move to opposite sides of the cell. They're just starting that process. The other thing that's starting to happen is that you can start to see what happens to these chromosomes. They start to condense a little bit. They're starting to coil up, tighten up. Here was a lot more spaghetti-like. Here they're starting to get a little more coiled up. So that's what happens in prophase. In prometaphase, we start to see some other things happening, like the nucleus starts to dissolve. So it starts to become fragmented. And also, we have these regions called the kinetochore. Now, the kinetochore, again, I'm going to spend more time on this in the next slide. I'm just kind of giving you a heads up right now. But the kinetochore is right at the location of the centromere in those cells. So at the chromosome's middle, where the two sister chromatids are attached, is the centromere. And then there's the structure called the kinetochore that's found at the centromere. Another thing that's happening is those spindle fibers that I talked about here that were just kind of being spun out. Those are microtubules, and they're being spun out of these structures here in the asters. Eventually, by prometaphase, those are going to be stretching all the way around the cell to each other. And some of those microtubules of the spindle fiber are going to actually directly attach to a chromosome. Some of those, though, they don't attach to a chromosome. Some of them just attach to other microtubules. So that's what's happening in prometaphase. Putting it all together with prophase and prometaphase, you got four basic things happening. The mitotic spindle is starting to form, and the aster or the centrosomes are starting to separate from each other. The DNA is starting to coil up. The centrosomes eventually reach the poles, and you have these microtubules either attached to the chromosomes at the kinetochore or to other non-kinetochore microtubules. The next phase is metaphase, and metaphase is super simple. Notice that the spindle fibers are completely formed. They're totally connected. Notice that that nucleus is completely gone. The centrosomes are all the way at each pole. And what the important thing about metaphase is meta is like in the middle. And what we have here is the metaphase plate. So those chromosomes are all lined up at the metaphase plate, right there in the center of the cell. One sister chromatid of each chromosome is towards the north pole, and north is arbitrary, just so you know. We usually draw it north and south pole. And then one of those sister chromatids from each chromosome is closer to the south pole. And that each of those sides has its own spindle fiber that's attached to it, its own microtubule that's attached to it. That's kind of how they get into the center, is those microtubules, those spindle fibers, kind of push and pull and tug till they all line up at the center at something called the metaphase plate. In anaphase, what we have is the separation of the sister chromatids. So important. Those sister chromatids that were together are now separated, and we call them now with the name daughter chromosomes. So this is a daughter and this is a daughter. And if you wanted to, at this point, you could count the number of chromosomes. So back in metaphase, there was 1, 2, 3, 4, 5, 6, right, with sister chromatids attached. But if you start counting daughter chromosomes, you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It looks like within a single cell, you've got twice the number of chromosomes. We tend to not count this very often, but it is in this one brief phase of anaphase where it looks like you've got twice the amount of DNA because those sister chromatids are separated from each other. In the next phase that we're going to talk about, we're going to combine telophase and cytokinesis. So this picture here is just showing you what's happening in both at the same time. Remember, cytokinesis is what's happening to the cytoplasm. So there's a bunch of stuff going on in telophase, including look at those chromosomes. They're much more spaghetti-like. So they're uncoiling. Another thing that's happening is the nuclear envelope is reforming. So you have this nucleus that's forming all the way around that whole set of chromosomes. And remember, in a human, there would be 46 of those chromosomes. In this cell, and there would be 46 chromosomes in this developing cell. Cytokinesis, we're going to spend some time on that. That's like this division right here where you're separating the cytoplasm into two separate structures. Let's take a closer look at the mitotic spindle. Remember the spindle, that's like this spindle fibers right here. The mitotic spindle is made up of microtubules and some other proteins. If you don't remember what microtubules are, you might want to go back when we were talking about structures of the cell and take a look at that. This mitotic spindle is very important because it controls the chromosome movement during mitosis. And remember that chromosome movement is pretty complicated. We go from being evenly distributed through the nucleus to being pushed and pulled in prometaphase to being completely lined up in metaphase, separating at anaphase. So this mitotic spindle has a lot of work to do. In animal cells, the spindle microtubules begins in this structure called the centrosome. And so this whole thing right here is a centrosome. And you can see that it's also, the centrosome here is also where this microtubules are kind of being spun out of. Now we used the word aster before on the previous slide. Remember when we were right here, we were talking about this term aster. That just means star. So it's the star shape. And you can kind of see that star shape right here when we're talking about the centrosome, these microtubules kind of coming out of that central centrosome in an aster or star shape. So the centrosome is made up again of the microtubules, and in animal cells we've got these things called centrioles. And the centrioles, there's a pair of them, and they've got some pretty specific structures associated with them, but there's two of them in animal cells. If we look even more closely at the mitotic spindle, one of the things that you'll have to know is that the centrosome itself duplicates during interphase. So I'm going to go back up. Again, I keep flipping around on these cells, but I'm going to go back up here and show you that in G2, that's the second of the gap phases, you have two centrosomes, 1, 2. And that replication actually does happen in interphase. Those are going to form these two centrosomes that are going to migrate to the opposite sides of the cell during prophase and prometaphase, and then do all their fancy stuff like grabbing onto chromosomes and moving those chromosomes around. Spindle microtubules grow from the centrosome as those centrosomes are migrating to opposite sides of the cell. And I told you about aster. This aster is this radial array of short microtubules that extend out of each centrosome. The spindle includes, again, the centrosome, the spindle microtubules, and the asters. Now, I've mentioned the word kinetochore before when we were looking at the steps for mitosis, and kinetochores are proteins that are assembled at the centromere of every single chromosome. And I grabbed this one off the internet because I kind of liked the way it was showing a very nice close-up of the kinetochore. So here's your sister chromatids 1 and 2, and right there at the centromere are these structures called kinetochores. And you can see that there are spindle fibers that do attach to the kinetochore. During prometaphase, some of the spindle microtubules, and we're going to call them kinetochore microtubules if they're connected to the kinetochore, some of those spindle fibers attach to the kinetochore and they move the chromosomes. So again, if the spindle fibers are attached to the kinetochore, we're going to call them kinetochore microtubules. In metaphase, the centromeres of the chromosomes are at the metaphase plate. And I like the way that this shows you the metaphase plate because it tries to show you in 3 dimensions. So think about, like, the metaphase plate isn't a single line. It's literally a plate that goes through the center of the cell. And this is in 3 dimensions, like these ones would be farther away from you in space, and these ones would be closer to you in space. So it's on a plate, not just a single line like an equator. An imaginary structure, this is midway through from between each of the asters, right? About halfway through that cell. Here's a close-up of the kinetochore, and there's the kinetochore microtubules, and you can see how this really looks like an aster right here, the centrosome spinning out these microtubules and attaching to the chromosomes and moving them towards that plate, that metaphase plate. Another quick look at the mitotic spindle. So when anaphase is happening, the sister chromatids are separating. Remember, we just talked about that. And what's happening is that the sister chromatids separate and move along kinetochore microtubules towards opposite sides of the cell. The microtubules are shortening by this process called depolymerization or depolymerizing. Remember, to polymerize means to add more and more subunits. So to depolymerize means to remove subunits. You could also call this hydrolysis. And this is happening at the end of the kinetochore. So if you look at this, you can see here, it might be hard to see which part's shortening. Maybe it could be shortening towards the aster, or maybe it could be shortening here towards the kinetochore. So what some clever scientists did was they marked the fibers, the spindle fibers, and then they watched to see what happened. Did the mark move closer to the aster, or did the chromosomes move closer to the mark? And what we see is that the chromosomes move closer to the mark, which means that we're eating away at the spindle fibers on this side. It's like slowly getting eaten away. So this happens when we have that depolymerization. It's kind of crunching off these little subunits of the microfiber or microtubules. So chromosomes are also reeled in by motor proteins. So we got some motor proteins coming along here and kind of pulling the spindle fiber through. Once it gets pulled through, then it breaks down. If we look at the non-kinetochore microtubules, if we focus on those for just a second, we can see that they actually start to push against each other to allow the cell to elongate. So here are the ones in purple are the kinetochore microtubules, and the ones in green are the non-kinetic core microtubules. And what happens is that, again, these push on each other, pushing the centrosomes farther and farther and farther apart so that the cell can stretch out and elongate and can eventually pinch into two separate cells. So if you think about it, if you really think about when cytokinesis starts to happen, it's kind of starting to happen sort of during anaphase when these non-kinetic core microtubules are pushing against each other, because that does allow the cell to elongate, and you gotta elongate before you can pinch in. So it's going to be fully elongated during telophase, and at that point, when it's fully elongated and you get this pinching in thing happening, especially in an animal cell, then once that's done, then cytokinesis is done, and all of that spindle fiber kind of just disappears. It disassembles. So we did just very quickly mention the idea that in animal cells you get this pinching in process, and that pinching in pinches in at this place called the cleavage furrow. And this whole process is called cleavage. And to cleave, like you think about a cleaver is a kind of a knife. So the cleavage furrow is the place where the cut is kind of happening. But it doesn't really happen like a cut. It's not really happening with a knife. We have this pinching in concept happening. And we have these proteins that are contracting and contracting and contracting until it's fully pinched in and two daughter cells form. Now, with plant cells, anything with a cell wall, that pinching in, that doesn't make any sense because that cell wall is much, much, much too hard. So we don't pinch in a plant cell. Instead, what happens during cytokinesis is that we get these vesicles that form along something called the cell plate. And as telophase is happening, the vesicles are forming all along here in a three-dimensional structure. And eventually, by the time cytokinesis is done, you have a full and complete cell wall, and that allows you to have two new cells. So the cell plate forms during cytokinesis in plant cells, and a cleavage furrow happens during cytokinesis in animal cells. Just kind of looking at what happens in a plant cell since the last time we looked at mitosis, we were looking at what was happening in animal cells. So just very, very, very similar. Like everything that happens in the nucleus is going to be the same. You got your prophase where the chromosomes are condensing. I didn't mention before that the nucleolus is beginning to disappear. That happens in animal cells as well. Then you can't really quite see the spindle fibers here, but they are starting to the formation process. During prometaphase, you really can see very clear chromosomes, and just remember that those are in the sister chromatid form. Here it is during metaphase on this metaphase plate, and notice that the entirety of the chromosomes aren't on the plate, but the centromeres are, and that's really important. You can also see right up here, you can see the asters there and there. During anaphase, those sister chromatids split apart. This is the same that happened in animal cells. Remember that our kinetochore microtubules are shortening and our non-kinetochore microtubules are pushing apart, but we don't get pinching in. And this is the key. We get this cell plate forming. Now, the stuff that happens in telophase is still happening in telophase. Like those chromosomes, you can't see them as easily, right? The nucleus is starting to form. The spindle apparatus is starting to disappear. All of those things are still happening, but we have a cell plate instead of a pinching in and cleavage furrow. Well, that's all that I have for you for the first part of Chapter 9. I've only divided Chapter 9 into 2 lectures, so stand by for Chapter 9, Part 2, where we talk about mitosis with regard to evolution, regulation, and cancer.